Hi guys, so today I'm going to help you out a little bit with your protein synthesis graphic organizer. So as soon as you open it, put your name at the top, and then we're going to talk about what is the central dogma of biology, really of protein synthesis. What a central dogma means is the main law of how things are made. So in this case, we have to start with our instructions, which is our DNA. And then our DNA is going to get copied, because remember, DNA cannot leave the nucleus, it's too fat. And it's going to get copied into mRNA, which is messenger RNA. And then that messenger RNA is going to take that information out and take it to the ribosomes, where we're going to make proteins. All right, it's all about making proteins. Your DNA is your instructions for making proteins, and your body is mostly made out of proteins. So let's go back over the structure of DNA, some of the characteristics. And the bases they have are A for adenine, T for thymine, C for cytosine, and G for guanine. And we should know our complementary base pair rules that A always pairs with T, apples in the tree, and T will also go with A. And then C always goes with G, cars in the garage. G with C, cars in the garage. Now, the type of sugar that your DNA is has to do with its name. So you have the DNA, and the D stands for the name of the sugar. And the name of the sugar in DNA is deoxyribose. All right, and that's the sugar that's in the backbones on the side along with the phosphate. All right, because remember, your bases are in the middle. So the full name of DNA is deoxyribose nucleic acid. So the name of the sugar is there. The function of your DNA, you should hopefully know that it is your genetic code. And what I mean by that is that it gives us our traits. So what are traits? Those are your physical characteristics, like the color of your hair, how tall you're going to be, your skin tone. All those things are determined by your genes on your DNA. Then we got RNA, DNA's skinny cousin. In RNA, it's very similar. It's half the size of DNA, which is why it can fit out of the nucleus. And there are some chemical differences. We still have A for adenine, but instead of T for thymine, we have U for uracil. And then we have C for cytosine and G for guanine. That does not change. So because of this U, it changes what pairs to what. And so these would be the bases in the DNA, because that's the template for your RNA. And in this case, if I was making RNA, A would not go with T anymore. Instead, it goes with U. T, though, however, I missed it. T would still go with A. All right, you have A's in RNA. And then the cars in the garage thing is the same. So C goes with G and G goes with C. That doesn't change. Now, RNA has a different name to it. It's an R in front, so the sugar's name is different. It's not deoxyribose anymore, and instead the sugar's slightly chemically different, and it's just called ribose. So it's actually a shorter version of deoxyribose, all right? And it's a skinnier version of the molecule. All right, so the function of this, well, there's different kinds of RNA, but the main function is to make the proteins. Right, to carry that code and facilitate the process of making proteins. So here's our different kinds of RNAs, so we don't need to address that right here. We're going to do this over here, and this is straight from your notes. So in your notes, you have what messenger RNA does, ribosomal RNA, what that is, and what transfer RNA does. So go into your notes and pick those out. I'm not going to give you the answers here. Now, if we're going to be comparing them, I will tell you that the two steps of protein synthesis that occur here, DNA to mRNA is transcription, mRNA to proteins is translation, and they go in alphabetical order, C before L. So both of these are processes to make proteins. All right, that's something they have in common. And then you're going to tell me here which one is, which I just actually went over up here at the top, okay? Then we have a diagram, a drag and drop diagram. So we're going to use these little guys right here that are on the side, which you can't see right now, but I will drag them over. So the first one up there is the nucleus of the cell. So if we're looking at this here, The nucleus is going to be over here, right, of your cell. This is your nucleus, and these are the nuclear pores. So inside there would be your DNA going through the process of transcription, okay? So this molecule that clearly has U's in it, it can't be DNA, and it's long, skinny, and half the size of DNA. It's only one side. That's going to be your mRNA. So you grab your mRNA label and put that over there. Okay? So going from the DNA in the nucleus to mRNA is transcription. Down here we have translation occurring. So when we're looking at translation, we have some codes we need to know. So the 3-letter code on this molecule, maybe we should name this molecule first. See here, this shape looks like a letter, looks like letter T, right? So this is your transfer RNA, your tRNA. So find your tRNA label, we'll label that guy, okay? And then the name of the code also has a T in the name, and that is an anticodon. So that's your anticodon. It's pointing there, those 3 letters. So 3 letters on the mRNA are the opposite of the anticodon. They are called a codon. And where this stuff goes and is occurring at this organelle called the ribosome. And the ribosome is made out of a certain kind of RNA, rRNA, which is, the R is for ribosome, okay? So the ribosome is the same thing as rRNA. Now, on your transfer RNA molecules, your tRNAs, they are carrying something. That's this black dot, okay? It's carrying something that you have to make the protein. It's the monomer of proteins. And the monomer of proteins are amino acids. Let me put that where you can see it. Okay. Let me put codon over where you can see it too. Oop, there we go. All right, so those are amino acids right there. And these amino acids will peptide bond to each other to create a protein. And that's what we have up here, right? This protein that is all these different amino acids bonded together, and once it pops off these tRNAs, it folds up. And the shape it folds up into is determined by the order of these amino acids. It tells it how to bond. So the order they're in, you make the shape. If the order gets messed up, you don't make the right shape, and then your protein does not work properly. And that's a problem, because you want your protein to work, right? All right, on the last page here, you're going to be using your code to find your amino acids for these different mRNA. So they're already mRNA, you don't have to flip them. You can tell they're mRNA by the U's. Okay. Over here, you have to tell me how many amino acids there are. That's right in your notes, or you could physically count them, which would be awful, but you could do that. And then there's a couple questions about different codes. So I'm going to do one of these with you, and I'll do one of these with you, and you guys are going to do the rest. So to find CAC, remember your first space is over here. Here's my C. That tells me I am in this row. Up at the top is A. Tells me it's in this column. And so where the 2 come together is in this box right here. All right, and then the C, there's 2 different ones in there. So I got to know the C tells me it's the second one, which is this one. And you only have to do the first 3 letters, so H-I-S. So you're going to go in here. Ooh, no, go there. All right, we're going to go in here and we're going to put H-I-S. H-I-S. And that's all I need. I don't need the whole word. If you want to, fine. We usually abbreviate them. So you're going to do that for each of these bases. Now to go backwards, it says list the 6 codons for the amino acid serine. So you got to find serine to begin with. And here it is up here. But it says 6 and it's only 4. Well, if you look over here, there's 2 more. Okay. So what code would give me this? Well, my first letter that could get me in that area would be U, right? The second letter would have to be C, because that'll get me in this box. U, C. And then the last letter, well, they're all serine, so it could be any one of these, but I'm going to do U, C, U. And that would be one of my codes, all capital. It would give me serine, and that's how you do that. So you're going to do that for all the rest of them. So I hope that helps y'all. When you finish, make sure you turn it in. Bye.